Hello everybody. In this video, we are going to have a look on lac-operon system. In prokaryotes, the regulation of gene expression is quite common and it occurs through a process of protein synthesis. It is implemented either by induction or repression by the transcriptional or translational level. Most of the bacterial genes of related functions are grouped under the control of a single promoter. The genes are transcribed into single mRNA. Polycistronic bacterial genes are transcribed together is known as operon. A well-known example for this operon system is lac-operon. The terminology lac refers to lactose molecule. Generally, bacteria digest glucose molecule for energy source. In case of absence of glucose, the E. coli turns on their genetic machinery to metabolize lactose molecule. The synthesis of glucose is easier when compared to lactose. If glucose is absent, it's time to turn on the production of enzymes to metabolize lactose into glucose and galactose. The key components of lac-operon system are promoter, means the RNA polymerase that first attaches for transcription. Operator, it is an switch on and off switch for transcription. The promoter block the RNA polymerase enzymes. Structural genes. So these genes that code for the enzymes to break down lactose. It has three combinations of genes, namely LAC-Z, LAC-Y and LAC-A. Regulator gene that produces a repressor protein by LAC-I gene. Let us see what's happening in the presence of glucose. If a glucose molecule is present in the gut of human being, the repressor protein binds with the operator and blocks the RNA polymerase to move towards the structural gene, and thus it inhibits the transcription. This process is known as switch-off mechanism. In the case of absence of glucose, the lactose is present. Here, the repressor protein should stop its function. In this case, the lactose molecule in the medium that alters and inactivates the repressor protein to prevent binding with the operator. Now the RNA polymerase enzyme is freely allowed to pass through the operator and promote transcription of LAC-Z, Y and A genes. LAC-Z produce beta-galactosidase enzyme that help to convert lactose into glucose and galactose, and now the bacteria can directly use the glucose. LAC-Y, a gene that produces permease enzyme which helps to increase the cell permeability. LAC-A produces thiogalactoside transacetylase enzyme, but unfortunately this enzyme has no function. So the lactose molecule will break down into glucoses and galactose molecules. Thus, the lactose molecule and the lactose metabolism happens finally, and the process is known as switch-on mechanism. Hope you understand the concept through this video. Thank you.